And reserve mobilization, dry matter partitioning, Ka ratio of rice seedlings in response to varying levels of salt stress. So we are looking into an early crop and salt stress as the first contribution. And I would like to ask Christian to share his screen and give his presentation. Everybody else, please switch off the microphone. Very good. Christian, your mic is off. Please switch on your mic.
Sorry about that. I got a little confused. Okay.
Here we go.
All right. I guess I'll start then. So Professor Asch already did a wonderful job introducing me. I'm a PhD student here at Hohenheim at the Management of Crop Water Stress in the Tropics and Subtropics at the Hans Rutenberg Institute. And it's my pleasure to discuss my presentation, of whose title you've already heard from Professor Asch, but I guess bears repeating. And that is reserve mobilization, dry matter partitioning, and sodium uptake of rice seedlings in response to varying levels of salt stress. So to start off with, I just want to outline salinity as an abiotic stress, much of which you already know. As you probably imagine, salt is bad. And in general, over years, through different crops, it's been shown that salinity reduces plant growth, development, and yield. Most crop species are glycophytes, and this is why this is a problem. That means they are salt sensitive and need to be optimized for continued cultivation on increasingly salinized cropland. Where does salinity come from? So this is both the product of natural and man-made processes, such as irrigation and climate change. And speaking of which, and of particular interest to me, because of my experience in Vietnam, that with sea level rise, there is an increasing saltwater intrusion in coastal areas. So we're talking about most of the major delta regions, thinking of Bangladesh or the Mekong Delta, which is increasingly leading to crop losses and subsequent migration of farmers. Moving on to the physiological element, how does salinity stress actually work? So actually it is a relatively complex phenomenon in that it is not just one, but two different processes that are happening over a period of time. So the first one is osmotic, in which the salt concentration is increasing around the roots, which lowers the water potential. And after reaching a threshold, typically will reduce shoot growth. And this holds for several species. After a period of time, this actually leads to an accumulation of sodium and chloride. As they're dissolved in water, this typically follows the transpirational stream within the plant. And as they're deposited in the most active leaves that are transpiring, this means that older leaves have toxic accumulations of sodium and chloride, reducing photosynthesis, and over time, eventually just killing the plant as the leaves senesce, eventually leading up to the youngest leaves. So if we're looking at crops and then saying, okay, we have salinity stress here in our first graph, we have a shoot growth rate. This is just representing the theory which I just discussed, where as you can see, there's a steep drop in performance in terms of shoot growth rate once the plant is introduced to salt. And as you can see, there's an initial period, a plateau, as the plant experiences osmotic stress, followed by eventual death as it goes into the ionic phase. If you were to look at a plant and hoping for increased osmotic tolerance, then you can see the dotted line that you still have an ionic phase, you still have reduced growth, but it's able to handle that initial drop better. And then if we're just discussing ionic tolerance, as you can see here, you still have a steep drop that represents the osmotic phase, followed by just continued barely surviving over the ionic phase. Ideally, we don't want just one or the other, we want both. As you can see, that is able to withstand both osmotic stress and ionic stress over a period of time. Though again, it is a stress, so you will always have a reduction in growth. So moving on to actual rice, Rice is the most sensitive to salt stress of all the cereals, by which I mean wheat, mostly wheat and barley. And as an example of which, rice yield has been shown to decrease up to 50% at 6.9 decisiemens per meter. However, this of course does not tell the whole story, in that there is a significant amount of variation according to variety. So for example, indica type rices are typically more tolerant to salinity stress than japonica type varieties. And then of course, it also depends on development, which complicates the story even further, in the sense that a plant and its tolerance can vary over a period of time just by virtue of what development stage it happens to be in. And what has been discovered over time in rice is that it tends to be most susceptible to salinity stress in the seedling and reproductive stages, as shown by the illustrious authors below, one of which is Professor Ash here. And as we can see here, we have the life cycle of the rice plant very briefly. And as you can see, the red rimmed pictures are the areas within the life cycle that are most susceptible to salinity stress. So the seedling, and then finally, when the plant is in this reproductive stage. So keeping that in mind, we devised an experiment looking at the intersection between salinity, variety, as well as development stage. And in particular, we're looking at the seedling stage, or at least from germination until an undetermined point, in this case, usually 2 weeks. So towards the end of the seedling stage as it moves on and starts to tiller, in theory. And to look at this in greater detail, we devised 3 different salt treatments: 0 millimolar, 50 millimolar, 100 millimolar, with 4 different rice varieties. In this case, 3 indica and 1 tropical japonica, and some of which names you might recognize. So IR64, it's a high-yielding semi-dwarf classic, and is considered somewhat tolerant to salinity stress, followed by OM5451, which is a variety which we have collected from Vietnam. It's predominantly grown in the Mekong Delta, very popular variety, followed by R31785, also not typically grown that often anymore, but a high-yielding semi-dwarf, which is considered susceptible to salt. And then Ekong Pao, a tropical japonica landrace, which is considered tissue tolerant, salt tolerant. And again, this is found in literature, so that they have been commonly used within these types of experiments, except for OM5451, which was more related to our experiences in Vietnam. And I had 3 replications, and how the experiment worked was that there was destructive sampling every other day. So I germinated the seeds, and then after a period of 4 days, I would then selectively take a tranche of varieties and dry them out. And then afterwards, I would measure the dry biomass according to organ, so coleoptile, radicle, and kernel. And I would then also take all of the organs, the whole seedling, crush it, and with flame photometry, I would determine the sodium and chloride ion concentrations. And you can just see a brief graphic of the setup. So it's very basic. You have sand within seed trays, within larger trays that have salt solution. And here you can see a closer graphic of my little seedlings that are responding to some variety of stress and this would be 1 sampling date of 1 replication. So what we actually did, and the analysis that we applied to the resulting data, was hoping to capture the dry biomass fractions over time and how the reserves of the seeds were mobilized. And this is in part reflected through a series of linear regression analyses that were used to determine seedling vigor, according to a method devised by Ash et al. In 1999. And in particular, it relates to 2 regression lines, which I will have the pleasure of introducing to you in a second. And here I've taken, in this graph right here, a real example of, in this case, OM5451 responding or growing over time. And in the blue regression reflects the total weight mass over time. So as you can see, it starts at a certain point, which would be the kernel, and it dips as the kernel reserves are exhausted, and then slowly increases again as the plant begins to photosynthesize and develop carbon through that method, and not just rely on the seed reserves themselves. The red regression happens to be the kernel weight, and as you can see, it's very nicely decreasing over time as the energy reserves in the seed are exhausted as the plant uses it for growth. We've also applied a third regression, here we've named 2, which is simply reflecting the growth over time without taking into account photosynthesis. And based on these 3 lines, we're then neatly able to capture loss of respiration, the losses, energetic losses to respiration, the fraction mobilized for growth, as well as you can see in the very bottom, you have an actual structural fraction of the grain in which there's actually no energy that can be provided. So here we have an example of this analysis with IR64, and very nicely reflected here is the effect of salinity. As you can see, it increases the different treatments. You have 0 millimolar on the left at 50 and then 100. And just looking at the kernel weight loss, it's very nicely tells a story in that there's far fewer reserves being mobilized as salt concentration is increasing. And then also, interestingly enough, looking at the total weight, the total dry weight reflected in the blue regression, that it actually flattens out seemingly as the plant or the seedling is unable to move on to photosynthesis. It is either inhibited in some way or senescing at a certain point, which is certainly the case in the 100 millimolar treatment. And here I given a nice graphic in the sense that I show the blue areas, that is the organ development, because that is essentially the difference between the total weight and the kernels, the blue and the red. That area reflects the development of the organs, in which I mean the radicle and the coleoptile, which, as you can see very nicely, decreases as concentration of the sodium, sodium chloride increases. And then you can see the different regressions. The blue, the regressions referring to the total weight gain, total biomass being on the top, and then the kernel weight reflected on the bottom. And at a certain point, especially at 50 millimolar and 100 millimolar, actually these start to look relatively similar, other than in the sense of kernel weight loss, which is interesting in the sense that you have more reserves being used up and you have organ development, and yet there's actually still not very much productive growth. And to make a quick comparison, here we're looking at OM5451, which similarly, I don't know if I need to really go into as much detail in the sense that you already know what you're kind of looking at. Again, the blue, total weight gain, and then the total weight, I'm going to get it right eventually, total weight of the seedling. And there you have the red again of the kernel weight loss. And again, and if you were to make a quick comparison between the two, it's actually notable to indicate that IR64, and I can just tell you because I have knowledge of the numbers, is significantly mobilizing greater amount of reserves compared to OM5451, which is somewhat interesting. And then I have an interesting graphic, which looks as a bit of a head scratcher when you look at it immediately, and that is looking at, in the blue regressions, the sodium concentration over time compared to the relative growth rate, which is the columns on the bottom of the graphs. And if you look on the far left, here you have next to no salt concentration within the plant, which makes sense. It's 0, it's a control. And you have a decrease in relative growth rate in initial stages, but as the plant actually moves on and starts to increase from a day-to-day time step, then you have a positive RGR. Whereas the 50 millimolar, somewhat interestingly, that you have this massive this relatively stable amount of concentration of sodium to a certain point around 10 days, at which point there is an exponential increase in the concentration within the seedling. And the relative growth rate actually is somewhat stable in the very beginning, but then increasingly decreases over time towards the very end, where it becomes a bit more dramatic. Whereas 100 millimolar, you actually have far less concentration over a period of time, even though there is a general increase, which would make sense that there is some sort of equilibrium with the surrounding solution. And this trend holds for OM5451 as well. Again, you have In 50 millimolar sodium solution, you have, interestingly, a higher concentration within the seedling of sodium. And again, you have this initial phase in the first 10 days in which the RGR is actually increasing or relatively similar to that what you see in the control, followed by a stark reduction. And the reason for this would be that, again, you have these two phases. You have the ionic and osmotic, and the 50 millimolar simply does not have the same osmotic stress as compared to the 100, which means that the plant is operating more or less similarly as is in the control, followed by, over time, an accumulation, or toxic accumulation of sodium ions, which leads to the significantly decreased RGR over time.
You have one more minute, Christian.
Okay, so I just wanted to briefly show you that I did measure the allocation between organs, above ground and below ground, within the seedlings. And as you can see, that there was not a huge amount of differences actually between above ground and below ground, at least that were significant, which again indicates the lack of transpiration influence within these plants as they're so young. And to just wrap it up, there is an increasing sodium solution concentration, sodium chloride solution concentration inhibits reserve mobilization and overall seedling development. And sodium chloride uptake begins with organ development, not inhibition. There are varietal differences in terms of ion uptake and growth. Rice seedlings, interestingly enough, grown in 50 millimolar solution had higher concentrations of sodium chloride compared to those in 100 millimolar. And this reflects different responses to sodium chloride in solution over time. And where does this lead me for the future? And that is, I would like to actually increase the length of the study to fully capture the end of a reserve mobilization, in particular for the 50 millimolar solution, which I never had a chance to fully see. And I'd also like to be able to draw a starker differentiation between the effects of the osmotic and ionic components of salinity stress. And in that case, I would like to construct an experiment in which I relieve the salinity stress. And if it happens to be ionic stress, then in that case, it'll be much, the plant will not be able to recover. Whereas if it was purely osmotic, then it should be able to recover after experiencing salinity stress. And on that note, I just want to thank you very much, Water People Agriculture Scholarship, which has supported me in my work so far. Thank you. And these are my references. And above all, thank you for your time and attention. And I look forward to your questions.
Well, thank you, Christian. That was a tour de force through salt stress related issues. We are running already over time, but I still want to ask if there are any questions. I don't see anything in question and answers here. So anything from the audience in the Zoom session? No, I have one short question. You said that there is a structural share of kernel reserves that is not remobilized, and you gave this as 5 point something. I wonder how you did determine this, since none of your data points seem never to reach that line. So
actually, well, this is based on previous literature, and secondly, actually, I've looked at the fractionation within seeds based on prior research done by Erie and so on, in which they actually identified how much of the seed was taken up by which structures, and that tends to be between 25 and 30% of the seed is not mobilizable, so to speak.
Okay, and so you just basically for your experiments, you just determined it you just put it at that level and without determining it yourself.
Yes.
No,
yeah.
Fine. No, that's fully valid. Okay, thank you very much, Christian. A big hand for Christian from the audience. We have about 25 people attending to this session here. Now, our next speaker will be talking about rice again, and in this case, root architecture of rice as affected by phosphorus starvation and salt stress. So here we have the salt stress again, but an additional factor, and that is a lack of phosphorus. Phosphorus is often a limiting nutrient in particularly tropical and also rice production systems and legumes also. And so it is an important aspect here. And we are taking it from salt stress now to salt stress again, but an additional factor, phosphorus, and we are coming back to phosphorus then in our third talk. So I would like to ask the ATSAF host to now play the video because our speaker cannot be with us. Here we come.
The name of the present work is Root Architecture of Rice as Affected by Phosphorus Starvation and Salt Stress. This work is a result of a collaboration between University of Havana and University of Rostock. Salt stress is one of the most harmful environmental factors affecting production of crops. In Cuba, about 15% of the agricultural lands are affected by salinity. Plants exposed to high salinity also suffer from nutrient deficit, like phosphorus. It is known that salinity and phosphorus availability can modify the root system architecture, but there are not too much information regarding their combined effect on root system architecture, especially in rice. That's why our aim was quantify the variation on root system architecture of two rice genotypes, Inca LP5 and Perla de Cuba, independence of salt level and phosphorus availability in mined rhizotome under controlled conditions. Seeds of Inca LP5 and Perla de Cuba rice genotype were placed in mined rhizotome in 4 different controlled conditions. Condition with Yoshida solution with normal concentration of phosphorus 10 ppm and low phosphorus concentration 1 ppm with normal concentration of sodium chloride and high concentration 50 millimolar of sodium chloride. After 20 days of growth, the root system was carefully placed on a scanner and analyzed with HERE-root-free software. This software measures extent, size, and distribution tract of the root system. Also, plant biomass and phosphorus uptake were measured. First, we would like to show the variation of dry weight in Inca LP5 and Perla de Cuba genotype with different conditions of phosphorus concentration and salt concentration. Inca LP5 genotype had a significant decrease in presence of saline stress, but not in presence of low phosphorus concentration. The same occurred with Perla de Cuba genotype. There was a reduction of dry weight in presence of high concentration of salt, but not in low concentration of phosphorus. The biggest decrease in dry weight occurred with saline stress and low P concentration. Korsklo-Sima et al. In 2012 have similar results in two barley genotypes under P starvation and saline stress, with an extreme reduction in biomass under saline stress. Also, Tang et al. In 2019 showed that the lowest biomass value in maize were with the combination of salt stress and P starvation. This decrease in plant biomass is also observed in the reduction of root system architecture. This image shows the root system of Inca LP5 genotype under 4 different conditions. And the present table shows 3 extent traits. These extent traits are network depth, network width, and network convex area. Any of these traits was affected by low phosphorus concentration, but was affected by salt concentration, high salt concentration. As you can see here, network depth was not affected with low peak concentration, but was affected with high concentration of sodium chloride. And the biggest reduce was with the combination of low peak concentration and high salt concentration. The same occurred with network width and with the network convex area. Here we can see the three distribution traits. The only distribution traits that was affected by both peak concentration and salt stress was maximum number of roots. But network length distribution and specific root length was not affected by concentration of phosphorus or concentration of salt. However, when we analyze the side traits, network perimeter, network surface area, and network length, we can see that all of them were reduced in the presence of P starvation or in the presence of saline stress. Perla de Cuba genotype was a little bit more affected. All the extent traits except network depth were reduced by peak starvation, and all of them for salt stress. We can see that network width was affected by low concentration of phosphorus and more affected by high concentration of salt. In general, the distribution traits were more affected by salt stress than low concentration of phosphorus. Only maximum number of root was reduced by peak starvation. And finally, like in Inca LP5, in Perla de Cuba genotype, the perimeter, surface area, and length were reduced significantly by both stress, peak starvation, and high salinity. In general, we can see in this picture that Perla de Cuba was more affected by peak starvation and salt stress than Inca LP5, especially in root size and root extent. In both genotypes, saline stress affected more than peak starvation. These results are similar to funded by Kawa et al. In 2016. They show that both salt stress can change the root architecture with a dominant effect of salinity in Arabidopsis. Also, Tang et al. In 2019 show a decrease in root length and root surface area in mice under saline stress and phosphorus availability. And finally, as expected, we had a significant reduction of reuptake, phosphorus uptake in Inca LP5 and Perla de Cuba, principally with salt stress and both stresses. With all these results, we have the following conclusion. Plant dry weight was decreased by salinity and peak starvation, but salinity had a stronger effect, especially in Inca LP5 genotype. Saline stress and phosphorus starvation affected the root system architecture in both rice genotype, but the saline stress affected more than the phosphorus starvation, especially in Perla de Cuba genotype. Plant phosphorus uptake was decreased by salinity and phosphorus starvation, but salinity had a stronger effect, especially in Perla de Cuba genotype. We would like to thanks to BNBF program and the project Innovative Fertilizer and Resource Efficiency in Agriculture. I would like especially thanks to Brigitte Klaus and Marcel Ackermann for their support in lab.
Okay, thank you very much for this contribution. Unfortunately, we cannot have the presenter live with us. And I think also that we cannot really make her answer questions as far as I can see, because I don't see any questions here. I think if there are questions for this presentation, they should be written into the question and answers and maybe then answered later by the author when there is a connection directly. So that was an interesting talk. I still have a couple of open questions, but maybe one that Alejandro can also address later, because I always thought that P deficiency leads to an increase in root length because the roots are basically looking for phosphorus. But here in these results, we saw that actually P starvation didn't really elongate the roots, but rather shorten them on top of it, which could be maybe an effect of the hydroponics that were used instead of using soil. I don't know. But anyway, I think we keep that question in mind, but we will not answer it now. But we will continue with our session here. And the next speaker will be Alejandro Peters from Venezuela. And he is talking about phosphorus acquisition efficiency in rice, and asked the question if carbon supply limits root growth under extreme P deficiency. So we are coming back to P deficiency here, same as in the talk before, and maybe here we find an answer why the root growth is actually so much limited. So Alejandro, the floor is yours.
Okay, first of all, thank you very much, Volker. First of all, can you all hear me?
Yes.
Okay, good. So yes, so what I will start is with a short introduction, because phosphorus, as we all know, together with nitrogen, is one of the most limiting nutrients for crop growth and yield. And also a large fraction of arable land has soils with low phosphorus availability, which makes agriculture highly dependent on phosphorus application. On top of that, soils with low phosphorus availability are mainly located in South America, Sub-Saharan Africa, and Southeast Asia, where farmers with low income are high in numbers. So obviously that makes the need to develop a strategy to produce or to breed for phosphorus efficient rice. And to stay all of us in kind of a constant context, I would like to show a few general effects of the impacts of phosphorus deficiency, which are general to plant. Basically, phosphorus deficiency decreases biomass and leaf area and consequently yield, also produces changes in biomass partitioning, and also in phosphorus distribution within the plant and within the cell, and also decreases photosynthesis, and as a consequence, it also alters carbohydrate metabolism. So the breeding strategy so far that had achieved more success is the breeding for phosphorus acquisition efficiency, which is basically the capacity of the plant to take up phosphorus from the soil or from the nutrient solution. Genetic variation, this is a graph where we can see here phosphorus uptake, and each spot is a different variety. So genetic variation in the capacity to acquire phosphorus from the substrate has been identified. And the other thing that has been identified is that this capacity for phosphorus uptake is very closely related to root weight, to root dry weight in terms of grams per plant. So the facts in kind of a small summary is that high phosphorus acquisition efficient rice varieties produce more root biomass on the phosphorus deficiency, which means that more carbon is required to sustain root growth. However, these occur on a background in which photosynthesis is impaired on the phosphorus deficiency, therefore decreasing the carbon availability for general growth, not only for roots, but also for shoots. So obviously these facts raise some questions, like for instance, is really carbon limiting for root growth? And where does the carbon comes from to feed the roots for increased proportional of root growth on the phosphorus deficiency, and whether this carbon is the produce of remobilization of previously stored carbon, or is the product of transport of current or recently assimilated carbon through photosynthesis. So in order to answer some of these questions, we designed a relatively simple experiment. We grew 4 rice genotypes, which were very different in the capacity in phosphorus acquisition efficiency, according to values reported in the literature. We germinated them, we transferred them to hydroponics Yoshida solution containing very low phosphorus concentration, and after 10 days we transferred half of the plants to a solution containing a normal or high phosphorus concentration, whether the other half remained in the low phosphorus concentration. So what I'm going to show you next is some of the results. We will start with the phosphorus status of the plants. Each color here, it represents a different variety, and the panel on top is a phosphorus concentration. The panel in the middle is phosphorus use efficiency, and phosphorus content in the lower panel. Within each graph, the box plots on the left are high phosphate plants, and the ones on the right are low phosphate plants. So the general results that we found in phosphorus status of the plants is that, as expected, phosphorus deficiency decreased massively phosphorus concentration, increased phosphorus use efficiency, and decreased phosphorus content. No differences in shoot phosphorus concentration were observed on the low phosphate treatment. Okay, differences in phosphorus status among varieties were more evident in roots, because we can see here that in shoots, not the phosphorus concentration or phosphate use efficiency were not statistically significant among varieties, whereas in the roots, the story is a bit more interesting. When we analyze the dry weight of each of the varieties, the structure of the graphing is the same. So shoot dry weight, in general terms, phosphorus deficiency decreased total and organ dry weight, but the impact differed among varieties, as we can see here. Not all varieties were equally affected by phosphorus deficiency. And varieties DJ123 and Xanthi sulfate, which are supposed to have the largest phosphorus acquisition efficiency, had the largest root biomass on the phosphorus deficiency amongst all the varieties compared. And the other interesting result here is that high phosphorus acquisition efficient varieties increased root to shoot ratio to a larger extent compared to IR64, which is, as Christian said at the beginning, is a kind of universal check and is also a low phosphorus acquisition efficient variety. We analyzed the gas exchange, specifically for the photosynthetic rate, and we found that it was inhibited, very importantly, inhibited by phosphorus deficiency. And again, the impact was different among varieties. So we've got an important genotypic by environment interaction, which is very encouraging when we look at breeding, because we've got genetic variation in the response. So these two graphs are basically the same. These are just plants measured when the nitrogen solution, the phosphate concentration was changed 70 days and 70 days after we measured the plants, and the other graph, plants were measured 25 days afterward, with essentially the same result. When we looked at the carbohydrates in the leaf blades, we observed that phosphorus deficiency decreased sucrose concentration, and the impact seems to be, again, dependent on the variety, at least when plants are sampled 20 days after the treatment. But the starch showed the opposite trend as sucrose, with a neat increase in starch concentration in the leaf blades in low phosphate grown plants compared to the control plants. When we looked at carbohydrates in the roots, I mean, in carbohydrates where they are needed in order to stimulate root growth, there were differences between the leaves and the roots. For instance, in roots, phosphorus deficiency also decreased both sucrose and starch, suggesting that carbon could be limiting in roots. The final panel here graph is the sum of root starch and root sucrose. That's what I call root total carbohydrate. So we've got not only a difference in sucrose and starch, but also we've got a decreased availability of total carbohydrates in the roots on the phosphorus deficiency. When we try to relate some of the process, try to look at some causes that could help us to explain the results, we saw that we have good evidence that the carbon seems to come. The carbon provided to produce root biomass seems to be provided by photosynthesis. That at least on the phosphate deficiency. If we see here in the upper panel, this is the assimilation rate plotted against root dry weight, and there is a very close relation between assimilation and root dry weight, and the strongest relationship and linear relationship was found during on the conditions of phosphate deficiency. So the other question, is carbon limited for root growth? Apparently, yes, mainly starch, as we can see, a very strong linear relationship between root starch concentration and root dry weight. A bit more complicated scenario is related to sucrose, but that can be complicated by the fact that not only sucrose being synthesized in the roots, but also sucrose being transported from the shoots to the roots. So that can complicate the relationship between what we actually measure as sucrose concentration in roots and the actual dry weight. So to finish off with some conclusions and outlook, we can say that under phosphorus deficiency, carbon seems to be limiting for root growth, but the magnitude is variable. The magnitude of the impact depends on the variety we select for. Maintenance of photosynthesis, therefore, and understanding of its regulatory mechanism on the phosphorus deficiency should be considered in breeding for phosphorus efficient rice, provided the close relation that we just showed between assimilation rate and growth and root dry weight, particularly under low phosphate conditions. On the other hand, we've got that genotypic variation was observed in photosynthetic response on the phosphorus deficiency, which was independent of shoot phosphorus status. If you remember, the phosphorus concentration in the shoots on the low phosphate was low compared to high phosphate, but there was no difference between varieties, whereas the photosynthetic response was dependent on the variety on the low phosphate. So that's suggesting that genetic differences in the regulatory process of photosynthesis on the phosphate deficiency. This indicates that the difference in photosynthesis on the phosphorus deficiency are not due to differences in the intensity of the phosphorus stress. As I was saying before, rather suggest genotypic differences in regulatory process, and obviously that makes necessary to look at the phosphorus internal phosphorus use efficiency, and not only at the phosphorus acquisition efficiency, in order to produce a really phosphorus efficient rice genotype. And with this, I finish my presentation for today. Thank you very much for your attention. I would also like to acknowledge the funding of the Baden-Württemberg Foundation for Persecuted Scientists that funded me for the last 2 years, and the University of Hohenheim and the group 49G for giving me all the chance and the opportunity to work in the facilities. Thank you very much.
Thank you, Alejandro, for this talk. You can stop sharing your screen, please.
Yeah.
I'm not sure if our questions and answers really function, or if these people out there just don't have any questions, because I don't see any questions coming up.
Yeah.
In the question and answers in the Woover tool. But there was a problem this morning already. I don't know if that has been solved. But anyway,
Okay.
that was a really nice talk. And there's one thing that came to my mind when you were presenting the relationships in starch and glucose relative to root growth.
Sucrose.
Sucrose, sorry. That, you know, when something has to grow in plants, it normally takes energy, and energy is normally provided in terms of phosphorus. So
Yeah.
ATP is turned into ADP in all these kind of regulatory processes. And starch is actually a storage organ, so a storage component.
Yeah.
I was wondering if you have a reduced root growth, but you have a functional photosynthesis more or less, and the root is not growing. So that could also be that there is carbon, yes, but the carbon cannot be processed because there's not enough ATP. And so it is actually not the carbon that is limiting the growth, but it's actually P is limiting the growth. Or do I get that wrong?
No, yes. I mean, PI is definitely limiting, could be limiting growth, and it's also limiting photosynthesis. The thing is that photosynthesis is so important for growth that, I mean, the whole system tries, regardless of the environmental conditions, tries to be kind of supported and be homeostatic regardless. And one of the things that happens is that, for instance, you've got the decrease in sucrose, and that means that less PI goes back into the chloroplast to regenerate RUBP, so photosynthesis can still go, but the increase, every molecule of starch that is formed releases actually PI back into the chloroplast. So trying to keep the system going. So the accumulation of starch, at least in the leaves, can be seen in two ways. One can be that you could have a kind of a storage of carbon for times when things go better for the plant, but also that accumulation of starch might be a way of keeping carbon going through the system because of the release of PI into the chloroplast for every mole of or molecule of starch which is formed. So actually, I was very kind of ambitious to try to put those questions, because I don't have very definite answers to those questions. And with these results, I can't say yes or no. What I'm showing here is that there is evidence that there should be some limitation, and we should also look at some of the mechanisms associated to be more certain of what the actual scenario is. Because on the other hand, statistical relationships don't show causal effects. It just shows that two things that vary in the same direction or opposite direction. Doesn't tells you whether which the mechanisms are behind that relationship, and that's what we would like to find out in the future.
Okay.
Yeah.
Actually, Woover seems to be working because I just got a question here from Thomas Hilger. He is asking, under natural conditions, there are bacteria and fungi that promote plant growth. Would this affect your findings?
Probably yes. The thing is that particularly in rice paddy fields, these microorganisms, for instance, the mycorrhiza are very limited in growth because the aerobic and anaerobic conditions. So not only in low oxygen, but also in terms of in any soil that is being used for agriculture for a long time, the dependence of the in general nutrients availability on microorganisms is very limited. because the massive input of fertilizers that are being used that somehow inhibit those microorganisms. So in theory, yes, they could help, and in natural environments is pretty common. The impact of mycorrhizas and so on. But in agricultural systems, at least in intensive agricultural systems, the impact could be limited, unfortunately.
Okay. Thank you very much, Alejandro, for this for the discussion. There are no further questions, but also we have to continue with our session so that we're not running too late. We are now moving away from rice. That was a good story on rice and phosphorus and so on. Now we come to other limitations, and we are coming to basically water deficiency, so water drought and water use efficiency and its effect on photosynthesis, but this time not in rice, but in a neotropical oilseed palm, and this Acrocomia aculeata. The writing here is quite small. And this will be presented by Katrin Meyer, who's also from Hohenheim, and I'm looking forward to your talk, Katrin. Sorry, I have to click a lot here because I have many windows open. So the floor is yours. You can share your screen, please.
So I hope you only see the PowerPoint, no? And not all the rest of the Zoom. So yeah, good afternoon. So I'm Katrin Meyer, also from the University of Hohenheim. And I will talk about and show preliminary results of how drought affects photosynthesis and water use efficiency in the oilseed palm, Acrocomia aculeata. So I will just dive in. And as we are talking about an oilseed palm, so of course we are talking about vegetable oils. So vegetable oils are one of the most important industrial commodities today and known a fast-growing demand. So this demand is mainly covered by the oil palm and the soybean, as we can see in the graph. And we all know the issues with the fast expansion of their growing areas in the humid tropics. Especially the optimal growing regions of oil palm are quite restricted to the humid tropics due to its low tolerance to drought. So that's the cue for Acrocomia. Acrocomia species are endemic to the semi-arid regions of South America. And it is an oilseed palm which is an underdeveloped crop, which shows a tolerance to drought and is considered a good candidate as tree component in intercropping systems. So which could make it a good alternative to oil palm in the semi-arid regions of the tropics and subtropics. So also the use of the fruits is very versatile, and also of the harvesting and processing residues. So the primary products are, of course, the oil, so ones from the fruit and also from the kernel. And also, for example, the oil cakes can be used as flour for baking or animal feed. As already said, we are studying the effect of drought on Acrocomia, and drought has an impact on a huge variety of processes in the plant, affecting plant growth and development. Also, drought is a limiting factor to crop expansion into new growing areas. The plant response has a wide range of levels from immediate to long-term effects. So the immediate response is closure of the stomata and consequentially the decrease in the photosynthetic rate. In the long term, drought results in a decrease in biomass accumulation and yield. So regarding Acrocomia, the questions are how the leaf gas exchange and photosynthetic rates are affected in the dry season and how it is the impact on different ecotypes. So the aim was to assess the stomatal conductance, photosynthetic rate, and water use efficiency in Acrocomia ecotypes coming from different climatic provenances. The study was conducted at the Macaúba germplasm bank of our partner university, the Universidade Federal de Viçosa, in Araponga, Minas Gerais, in southeastern Brazil. 5 ecotypes were selected based on the climatic provenance all over from Brazil. As we are speaking of climate, here we see the rainfall and the soil moisture content during the first period of the study, which was from April 19 to February 2020. And as we are in the southern hemisphere, the winter, which is the dry season, is from April to October, whereas the other half of the year is summer and the rainy season. The sampling dates are once end of September, so after 4 months of almost no rainfall last year, and when the soil moisture content was at the lowest. The second sampling date was end of January, so in the second half of the rainy season, where soil moisture content found its peak already. So what we measured, first the photosynthetic parameters, so photosynthetic rates, stomatal conductance, transpiration using an IRGA device. The water leaf status was determined by collecting leaf discs, weighting them, rehydrating, waiting, drying, waiting again, and then calculating. And finally, water use efficiency, which is an ecophysiological indicator and is defined by the accumulated biomass per amount transpired water. We are distinguishing two, so the intrinsic one, which is independent of the evaporative demand, and the instantaneous one, which is dependent of the evaporative demand. So to the results, first the relative water status. As we can see, it's lower in the dry season than in the rainy season. However, looking at absolute values, we see that even in the dry season, all ecotypes have an RVC above 88%, so clearly higher than the typical RVC around initial wilting of 60 to 70% in most plant species. The explanation for the higher RVC in this ecotype could be that it has thicker leaves with a waxy surface compared to the others, so reducing water loss. Next up are the photosynthetic parameters, so it's from above the bottom, photosynthetic rate, then transpiration and stomatal conductance in the bottom. And also here we see a depression in the dry season. So the ecotypes coming from drier regions are less affected than the ecotypes coming from humid regions, with the exception of this ecotype here, but we will come back to this in the discussion, so later on. So last we come to the water use efficiency. Here the term rural list, the higher, the more resilient is the ecotype to drought. They follow the expected pattern of a low efficiency in the rainy season and a higher one in the dry season. And first we look at the intrinsic efficiency, so the one independent of the evaporative demand. And in summer there are no differences between the ecotypes. However, in the dry season, some ecotypes have higher efficiencies. Surprisingly enough, also one of the humid adapted ecotypes, which is on the far right. We can also see that the ecotype which showed high photosynthetic parameters, so the one in the middle, has a low water use efficiency, so using more water to accumulate biomass as the others. Coming to the instantaneous water use efficiency, so the one dependent of evaporative demand, there are similar trends. So low in summer, high in winter, but we see that the most dry adapted ecotype, so the one completely on the left, is outperformed by the others in the rainy season. Whereas in the dry season, it is on the same level as the humid adapted ecotypes. However, looking at the efficiency difference between dry and rainy season, it is almost that level with the other dry adapted ecotype, with a difference around 3 micromoles, and the humid adapted ecotypes show a difference only of around 2 micromoles between the two seasons. So the discussion and conclusion, so nothing new. All ecotypes were affected by drought, whereas no impact was found on relative water content. The photosynthetic parameters were less affected in dry adapted ecotypes, with the exception of the one, where perhaps another climate pattern could be influencing, so further research is needed. What is efficiency? So the intrinsic one, we saw mixed trends. In the instantaneous one ecotype stands out. So we need to elaborate what influence the distinctive environmental factors have on the ecotypes. Natural differences could be observed between ecotypes, which is of course beneficial for the development of a cocomia to a crop through breeding. However, we saw some not straightforward trends, so the further investigation is needed. So this is on one hand, the further assessment of the data already collected, and on the other hand, the analysis of the 13C content as also additional field work. So with more tightly timed measurements of photosynthesis during seasonal change, and also measurements of the fluctuations throughout the day. But as so many other projects, we are also affected by COVID-19, and the analysis 13C is completely delayed, and the additional fieldwork is postponed to the end of next year, hopefully. With that, we reached an end. I want to thank all my colleagues and the workers from our partner, the Universidad Federal de Viçosa. Without them, fieldwork would have been very hard. And I also want to thank the Fonds National de la Recherche Luxembourg, who is funding this project very kindly. Thank you very much for your attention, and I hope there are some questions. You need to, you're muted.
Sorry, thank you very much for your presentation. That was highly interesting. I have lots of questions, but I think I will ask you then when you're back in Hohenheim.
Yeah.
But there was one question in the questions and answers, but that is actually not related to you. And it's also something we cannot answer here. I was told that we have 30 minutes left in the session so that we can go until 4:30. That is wonderful. So we have a little bit more time. I have a technical question that I'm still too curious now to leave it until you are back. And that is, how did you actually measure the photosynthesis in these leaves? Yeah. As far as I can see, they are 2.5 meters long, and they are feathery leaves and have about at least something like 55 component leaves in it. So how did you do that?
Yeah, it's so it's actually we use the method they are using there, which is actually cutting the leaves. So of the middle rib, so you take one of the whole leaf, you cut from the middle, a leaflet, make it in, and you have around a few minutes to measure until you have the depression because of the cutting down. Fortunately, a cocomia, it answers very fast in the machine. So normally you have it reaches the peak after 1 minute, 1 to 2 minutes. And then it's like the stable values. So that's quite positive, but it's more or less the only way to do it. Also because the palms are now between 5 and 9 meters high. So, and we take the second youngest leaf, so you actually can't reach. Yeah, it's the only way, is to cut it and measure as fast as possible.
It's an interesting challenge, really.
Yeah.
Do we have any other comments or questions? Christian looks like he wants to say something.
Yeah. I was wondering, have you ever thought about looking at the waxes, a cuticle analysis at the different ecotypes and whether the composition of the cuticle varied on the upper or lower parts of the leaves?
Yeah, I thought about it, but now in the frame of my work, of my project, I don't have the time. I also don't know if anyone is working on it, but I think it will be it's one of the projects which is in my head, because yeah, with acrocomia, there's still a lot to do, and yeah, but I was looking into it because it makes it also hard to count the stomatas, because this one has waxy leaves without hairs, and the others, they have hairs, so it's like in the leaves, so it's like quite different. And I've tried to find also why it is so different. Partly it's also the region, so it's already mixed up with another species of acrocomia, so you need to take care. So it's not probably not purely aculeata anymore, but there is the one from Paraguay is also mixing in it. And I forgot the name right now of that one. Yeah.
And I didn't understand as well for the different ecotypes. Is it in different ecotypes a shade tree versus a sun tree, or is it typically always in the same part of the forest?
No, the palms we use, they are simply from completely different regions of Brazil. And that's why I don't speak about genotypes, because we don't know if the genetic differences, but they are influenced by the areas where they are coming from. So the ones that you already can see in the fruits, so the ones from the northeast of Brazil have fruits which are very similar. So the oil you gain is very similar to oil palm, whereas the ones from the southeast and really from the west of Brazil, they have oils which are more like olive oil from how it looks, how it smells, and so on. Yeah, and okay. So you have a very huge difference. And of course, now I want to try if you can see it also in the photosynthesis, water use efficiency, and so on. Differences.
Yeah, so there would be definitely also different genotypes, but of course you do not know that.
Yeah, I don't know. That's why I'm speaking about ecotypes.
Yeah, of course, makes sense.
Yeah.
Yeah, thanks a lot for this discussion also. We are coming now to another crop again, and it is legumes, lentils in this case. We are staying with photosynthesis. So as we can see from these presentations, we are always coming back to photosynthesis, somehow growth, mobilization, internal processes. So that's where we are actually at the moment. And it's quite interesting that despite the fact that we are working on different crops, you're all coming back to the same issues. And of course, that has something to do with abiotic stresses. And these abiotic stresses are often imposed by climate change issues. And so the next talk will be focusing on high temperature. And it will not be given live. Tariqil Islam is with us, but he pre-recorded the video, and this will be now played into the session by our host in Witzenhausen. So please, can we have the video now? I can see the screen, but I don't hear anything. No tone. Or does anybody hear anything? It's just me?
It's not. If you look at the bar at the bottom, it's not playing.
Welcome you all to my presentation. I am Dr. MD Tariqul Islam from Bangladesh Institute of Nuclear Agriculture. The title of my study is Photosynthesis, Dry Matter Production, and Yield Performance of Lentil Varieties under high temperature. I can tell you some backgrounds and significance of this study. We know temperature is rising due to increasing concentration of carbon dioxide and other atmospheric greenhouse gases. The rise in atmospheric temperature causes detrimental effects on growth, yield, and quality of the crop varieties. Lentil is an important pulse crop with high protein content, has the potential capacity to combat nutritional deficiencies in developing regions and countries. It occasionally faces high temperature at its reproductive stage in February-March due to late sowing after transplanting almond rice harvest in Bangladesh and some parts of India. Effects can be made to increase area as well as yield of lentil crops by the use of temperature stress tolerant varieties. The objectives of the study are to assess the effects of high temperature 34 degrees Celsius on photosynthesis, chlorophyll content in leaves, dry matter production, yield attributes, and yield of the lentil varieties, and to find out temperature stress tolerant varieties. I can describe materials and methods. The experiment was carried out in pots during March 2017 to May 2018 at Bangladesh Institute of Nuclear Agriculture, Mymensingh, Bangladesh. 3 treatments: temperature, ambient 34 degrees Celsius at flower initiation stage and 34 degrees Celsius at pot filling stage of lentil varieties. In this study, 10 high yielding lentil varieties were used. The design of the experiment was complete randomized design with 3 replications. Temperature treatments were imposed separately for 7 days in controlled plant growth chamber. The nitrate reductase activity, chlorophyll content, and photosynthetic rate of leaves were determined during temperature imposed. Photosynthetic rate was recorded using portable photosynthesis system LI6400XT of USA. At harvest, total dry matter, seed yield, and yield related parameters were recorded. Finally, data were analyzed statistically and DMRT was done to compare the means. Now I can describe the results of the experiment. High temperature imposed either at flowering initiation stage or pod growth stage had detrimental effect on plant growth parameters. However, high temperature imposed at pod growth stage had more negative influence on plant growth parameters. Here we can see Binamosur 6, Binamosur 8, and Binamosur 10 produced longer plants. Binamosur 7 showed higher photosynthetic rate, whereas Barimosur 5 and Binamosur 9 produced higher seed yield and harvest indices. Here we can see that high temperature drastically reduced seed yield of all the varieties at any growth stages. Bina Musur 2 and Bina Musur 3 showed higher reduction in yield, whereas Bina Musur 6 and Bina Musur 8 reduced less yield under high temperature treatments. So here we can make conclusion that high temperature imposed either at flowering initiation stage or pod growth stage had high significant negative influence on plant growth parameters and yield. Temperature imposed at pod development stage had greater negative influence on morphophysiological parameters as well as seed yield. Among 10 lentil varieties, Bina Musur 6 and Bina Musur 8 had some tolerance to high temperature. This is end of my presentation. Thank you all for patience hearing.
Yes, thank you very much for this comprehensive presentation on lentil and high temperature tolerance. I must say I was quite surprised to see that 34 degrees during flowering has such a detrimental effect on all of your genotypes. I was really surprised. I thought lentil is a relatively tropical and dry area crop, so that there should be some inherent tolerance in the material already. So in the end, you conclude that you have some tolerance in 2 of your lines, but in the end, that was not that much of a tolerance, really. So what can you do? If you're facing climate change and high temperatures, can you actually in your system adapt or change the cropping calendar, or do you have to actually let go of lentils in the long run? What is your solution?
Okay, thank you for your nice question. I think this is 1 year experiment. I have to do this experiment again for final conclusion. However, lentil is a very sensitive crop. Although it has some drought tolerance, but it has a very limited limited drought tolerance, and salt, it has no at all salt tolerance. But temperature tolerance, you have seen in my results that all the varieties drastically reduced grain yield.
Yes.
Only 2 varieties showed less reduction in. 2 varieties have some tolerance to high temperature, 34 degrees Celsius is a high temperature. So I should repeat this experiment again, and if I get a very nice variety, then it could be cultivated there. Thank you.
Okay, thank you very much. I don't see any questions here in WUFA. How about the group here in Zoom? Do you want to? Do you have any questions to Tariq al-Islam and his study? That doesn't look like it. Well, I wish you luck with repeating your experiments and also with keeping lentil alive in Bangladesh. If ever I have the chance to come back to Bangladesh, I would like to see the Bari Nuclear Agriculture Institute. I wonder, what is this nuclear for? Do you really do like radionuclides, or what is the nuclear?
Radiation source from cobalt-66 We are using for radiation, and we are getting many mutants, and we have already developed more than 100 crop varieties
Amazing.
with this nuclear technique.
That's really interesting. So if ever I make my way back to Bangladesh, I will give you a call, and then you have to introduce me to this institute. I'm very curious.
Okay.
So thank you.
You are
very
welcome.
much. Thank you very much for your talk. Thanks to all of my speakers here. That was really a nice session. And I think each one of you contributed a very special aspect to abiotic stress research. And I hope for the audience out there, it became clear how complicated and how complex these things can be. And the devil is often in the details. And we are far away from understanding plant responses to abiotic factors. And if we take into account the genetic variety that's out there, we have still a very long way to go to actually have an answer to the looming crisis in terms of properly adapted plants, or in understanding the mechanisms that are behind it, so that we can actually make use of them. And that's why we are still doing a lot of research. And I hope that Sebastian Lesch is still listening to us here, and that he takes into account that research is not necessarily target oriented, and that we need some money to play around with in international relationships in order to keep the research going and come up with results that in the long run may have an impact on crop production and crop varieties. So with this, I thank you again very much for being here and for sharing your results with us. And I wish you a wonderful continuation of the Tropentag. And I'm ending the session now here with great hand to all of you. See you around. And thanks to ATSAF TT21. Very good service.